Okay, I was talking about that low fidelity DNA synthesis before the recording is cut up. So what that means is, yes, although DNA polymers can move along that translesion area so it can complete the replication, but those special DNA polymerases are error prone, because by reading through those strange templates area, it can make a lot of mistakes. That's what low fidelity DNA synthesis is. It can make a bunch of mistakes in the replication. Okay, so what's the benefit of making a bunch of mistakes, which is bunch of more mutations in the daughter strand? Okay, the benefit will be, let's say, so this is what is happening here. So that's a normal DNA polymerase 3, right, which is high fidelity polymerase. It has exonuclease activity. It cares how it replicates. Okay, and then normally when it faces like a lesion, like a dimerized covalently attached to T's, then it doesn't know what to do, so it just stalls there. Under the condition, if cells under a lot of stress, DNA polymerase number 3 falls up. Instead, these kind of special polymerases, polymerase number 4 and 5, they may join there instead of polymerase number 3, and then they can move along the lesion area. And how do they do that? By making mistake. Instead of adding maybe A and A's, it can just add some random nucleotide, maybe G and A, and just continue its polymerization. Okay, if it passes that lesion area, then regular DNA polymerase can come along and continue that area. So benefit of doing that is, instead of stopping the replication can continue. Okay, although there are mutations introduced. Okay, so the Y family polymerase can incorporate nucleotide in a manner that is independent of base pairing, meaning it's not always G to C, A to T base pairing on the daughter strand. That means it is making a bunch of mutations. Okay, and translesion DNA synthesis allows cells to survive a catastrophic block of replication. So replication block, so DNAs are replicated, then that's catastrophic, because cells will die because of that. But it introduces high level of mutation, like I said, and translesion polymerases are highly regulated, and their synthesis is only induced under the SOS response. That's a stressful condition where cells are, maybe exposed to UV light and things like that in response to DNA damage. So they are not always doing their job. It's only made available during the stressful condition. Cells are coping with a lot of stress and mutation. It's a matter of death or life kind of situation. Then these special polymerases are made available, and they can do complete their application, and they can make a bunch of mutations. Okay, in that gene regulation, RAC-C protein is important, because that is activated by the single-strand DNA resulted from DNA damage, and cleaves LAX-A repressor. So repressor is a controlling protein. It represses expression of the genes, like those polymerase coding genes and other enzyme genes that are involved in the SOS response. We'll talk about that in a minute. Okay, anyway, so these two the kind of regulator elements control the gene expression during this SOS response. So the main thing is, by introducing actually a bunch of mutations in the genome, that'll be like E. coli cell. So what is the benefit, especially in terms of evolution? The benefit is in evolution. So if we have a lot more genes that are brand new, okay, through these mutations, out of those newly mutated genes, there may be some kind of beneficial phenotype which may help these cells to cope with that stressful condition, and they may be better adapted. So they may be better surviving under this stressful condition. So that's the benefit of introducing a bunch of new mutations during this stressful condition. Okay, so that's how unique or neat this system is. Okay, so this showed you the SOS response. So so LAX-A and RAC-A, they are the key proteins that can control, especially LAX-A is a repressor that binds the promoter element, and that repressor means it turns off the transcription of many genes. Okay, but LAX-A repressor can be inactivated if it's a pain in the butt. If LEX-A binds with the REC-A, okay, and the REC-A can only bind with LEX-A when REC-A is activated. Then how is REC-A protein activated? REC-A is activated if it binds with single-strand DNA molecules that are maybe damaged by UV light and things like that. Okay, so when that REC-A binds with LEX-A, LEX-A falls off from the promoter site, then the genes that are repressed will be turned on, and those gene products will be those enzymes like polymerase enzymes, and then maybe repair enzymes that will deal with the DNA repair, DNA damages. Okay, so understand this SOS response. So it's kind of unique that UV can turn on the LEX-A repressed genes, because REC-A responds to the damaged DNA. It binds the single-strand DNA, and then REC-A, when it's activated, binds on the LEX-A, which is falling off. So repressor is inactivated. Then genes are turned on, and those genes will take care of the DNA repair as well as polymerization under this stressful condition. Okay. So that's what happens in E. coli. And how about mammalian cells? Okay, pCNA sliding clamp is ubiquitinated under these conditions. So you see that pCNA sliding clamp, ubiquitin is added to that. It's ubiquitination. So addition of ubiquitin peptide, that's what it means. Okay, so that happens under the high DNA damaging condition, like exposure to UV light and things like that. And once that is ubiquitinated, the sliding clamp recruits a translesion polymerase rather than normal polymerase. Okay, so they recognize the ubiquitinated this ring. So when that ring pCNA is ubiquitinated, then that recruits translesion DNA polymerase under the stressful condition, and that translesion polymerase will go through the damaged DNA and continue the replication. That's what it means. Okay. Translesion synthesis also use the mechanism of gap filling. See that? So to repair the gap resulted from the replicative DNA polymerase skipping over the DNA lesion. So what's happening is, so replication DNA polymerase is normal, and then it's ubiquitinated. So normal DNA polymerase doesn't know what to do, so it may just jump there. Okay, so pass over there, and then this translesion DNA polymerase will come along later on, and this fill up that gap that's left by normal DNA. It just jumps along and then do the filling, okay, and take care of the mistake. That's what it means. So basically what it means is, yes, eukaryotic cells do have similar repair system that deals with very stressful condition where a lot of DNA damages are being made, and then has to be handled during the replication. Okay, so that's the unique thing about this system. Okay, that's it for the lecture 6. Yeah, there are a lot of information, so I hope you can study detail about this. All right, that's it.